Dr. Jerolen Naidoo, thank you very much for joining us. And before we get into the kind of crux of what we want to talk about, maybe it's important just to set the scene for us. What is microbiomes?
A microbiome, in essence, just refers to a community of microorganisms. So these can be things like bacteria, fungi, viruses that inhabit a specific microenvironment. So we have microbiomes that exist, for example, in the soil, a separate microbiome that exists in the air, microbiomes that exist deep in the ocean in coral reefs. As NextGen Health, we're particularly interested in microbiomes that kind of co-inhabit the human body, and this is what's referred to as the human microbiome. So the human microbiome itself exists in multiple different spaces across the human body. So you've got a skin microbiome, for example, a separate microbiome in your mouth, one that exists in your lung, in your liver. And of all these sites, the gut, so the intestine, is actually the most abundant source of microbial diversity and microbiome in the human body.
The difference between microbiomes and genes in relation to the human body and your overall health, what role does it play?
So the central, dogma of biology is that we've got DNA, which serves as a blueprint. And from the blueprint, we then get an intermediate called RNA. And the RNA is then translated into protein. And protein, for the longest time, was thought to be the actual functional unit. That thinking has changed a lot. But when we refer to the microbiome as a whole, what we're referring to are basically all of the segments of DNA that encode these functional units. So a gene is essentially one segment of code within the DNA. And when we talk about a microbiome, we're referring to all of those functional segments of DNA encoded by all the different microorganisms that co-inhabit the human body. And so in terms of the relation of this to health, or human health specifically, it's very interesting. So we have slightly more bacterial cells in the average human body than we do human cells. So we're slightly more bacteria than we are human. And the human genome encodes 23,000 genes. But the totality of the genes encoded by the bacterial and microorganisms in the human body are in excess of 3 million. So they vastly outnumber the functional elements within the human body. And we know how important our own genes are to us from various knockout studies that show that if you remove certain genes, then there are drastic consequences for human health and cell function. The same is now true in terms of what we're learning about these microorganisms. They play a very essential role in maintaining human physiology and various different aspects of human health, and specifically our risk to disease.
So there is a direct relationship between kind of the gut microbiome, your health, your environment, and your lifestyle. There is a direct relationship.
Yes. So this is also a lot of recent studies have kind of uncovered this, and it's quite exciting. So we know in general, as a rule of thumb, that the more diverse your microbiome is, this generally correlates with better health outcomes and a healthier state in general. And the loss of diversity in terms of the microbes that are in your microbiome generally correlates with the onset of specific diseases or susceptibility to disease. And in terms of the environment, sorry, I know the question is a bit long, so there's a lot of information. In terms of the environment, what we've come to understand is that the environment is a huge driver of what our microbiomes actually look like. So it's you as an individual, your daily exposures, the things you do, the things you eat, whether you sit in traffic and how long you're exposed to traffic fumes, the air quality, how much of fermented foods or yogurt you eat, all of this kind of contributes to your unique microbiome profile.
So if geospatial factors play a role, it would look different if you were traveling or if you were living in a developed or developing country. Your microbiome system would look completely different.
Yeah, that's true. And there was an amazing study that was conducted in 2018 where they tracked immigrants from Thailand who were immigrating to the US. And what they did was they profiled their microbiomes at the point when they left Thailand. And then they profiled them again 9 months later after they had been in the US for 9 months. And what they found was that their microbiome profiles after 9 months actually more closely mimicked those of local Americans than they did their own microbiome profiles when they had left Thailand. And that new microbiome profile was actually correlated to a high risk for the onset of obesity and a loss in microbial diversity. And we know that these westernized diets, for example, in the US, are also associated with higher cases of obesity and risk for diabetes and diseases such as these.
So what causes your microbiome profile to change? Would you need to have a drastic change in the environment, or is it just a case of time and it starts mimicking the environment it's in?
Yeah, so that's a good question. So the microbiome, I'll start at the beginning a little bit. The microbiome is actually seeded in utero. So the developing fetus gets its initial microbiome from everything that the mum is eating and the mum is exposed to. And we now have come to understand that microbiome matures within the first 3 years of life. So at 3 years of age, an individual has already established what will become their core microbiome, and that stays with you generally throughout your adult life. So you have a core that exists, and around this you have what's a potentially variable microbiome, and that can change more drastically in terms of acute exposure. So if I ate something very foreign to me for like a couple of days, that might shift my variable microbiome, but my core would still essentially remain similar. But when you start doing long-term exposures to things like changing your diet, so for example, if I decided today that I wanted to become a vegetarian, my microbiome in 9 months, both the variable and the core, could have shifted now slightly. Other things such as chronic medication we found can also change the microbiome. And you think about it in terms of when you get sick and you go to the doctor, 9 times out of 10 you're getting antibiotics. Antibiotics, by their very nature, are designed to remove all microorganisms, or bacteria specifically, right, from the human body. And some of them are very non-specific. So what happens is that you get kind of a harsh reset on your microbiome. And then what needs to happen is that over time, because you're living a similar lifestyle than you were before, gradually your microbiome returns to normal, and that's what's called homeostasis.
Doctor, why is it important for us to have a broader and a deeper understanding of this South African profile when you talk about microbiomes? And also, how would understanding those profiles help us?
Over the last decade, there's been a tremendous increase in microbiome research, and this has really been catalyzed by the availability and the reduction in the cost of next-generation sequencing technologies. And what we've come to understand is that the microbiome really exists as another pillar of precision medicine in terms of what makes an individual unique and how that uniqueness then translates to clinical outcomes and risk for disease. So a couple of studies, and there are numerous ones, but I'll just highlight a few that are kind of interesting. So 2 cancer treatments, for example. One is cyclophosphamide, which is also out of interest, the #1 prescribed cancer treatment at Groote Schuur Hospital in Cape Town, which is most South African public hospitals have access to this drug. They've now found that whether the drug is effective or not as an anti-cancer treatment depends almost completely on how healthy an individual's microbiome is. So the way the drug actually works is that it perforates little holes in your intestine, and then what happens is that the bacteria actually leak out into your blood system, and the leaking out of that bacteria actually galvanizes the host or the human immune response against the cancer. So depending on whether you have a healthy gut or an unhealthy gut, the drug may be effective for you or completely ineffective. And the same has been shown to be true for groundbreaking immunotherapy for cancer. So PD-1 antibody treatment is literally the cutting edge cancer treatment, and it costs, I think, around 1.2 million per treatment course. And they found that, again, the microbiome is such a huge determinant of whether this will be successful or not. And that's just cancer treatment. There's also things like disease risk profiles where we've been able to find out like the microbiome is very strongly linked, and diversity of the microbiome, to diseases such as asthma, your susceptibility to infectious diseases like TB, HIV. All of these things are very, very closely linked to the microbiome, but it's still an emerging field, and we're learning more every day.
Where Africa and South Africa's position from the amount of health issues and diseases that we have, How would a microbiome digital platform kind of solve those health issues?
We do have a lot of problems at home. And I think recently South Africa was actually ranked as the unhealthiest country in the world by the Indigo Wellbeing Index. And that looked at a couple of different factors. One was government expenditure on healthcare. The other was things like risk of diabetes, obesity, cardiovascular disease, depression. And interestingly, all of those diseases that I just mentioned have been strongly linked to the microbiome. So in studies conducted, for example, in Europe and the US, so far we've seen that the microbiome can actually be causal to some of these diseases and the onset of these diseases. And as we discussed previously, there's a very specific kind of impact of geography on understanding how the microbiome relates to disease. And this knowledge is very much lacking in terms of South Africa and the African continent as a whole. So the onus really is on us as Africans to drive this research for our own benefit. So part of where I think a digital platform may be really useful is that it will provide us with essentially a novel health metric in terms of the microbiome, and using it as a metric to be able to evaluate what is the general health of the people within our population. And the beauty of it is that it's an early warning system. So the microbiome, because it's slightly malleable, can actually respond very early to the onset of disease as opposed to much later on. So the idea is that by picking up these changes in the microbiome early on, we can actually intervene before disease becomes too serious.
Now, we know that the CSIR has a microbiome mapping initiative. What does that entail? That sounds so futuristic to me.
So the CSIR Microbiome Mapping Initiative is essentially a pilot project that's been set up within the Next Generation Health Cluster. And the idea is for us to basically set up the infrastructure and the technical know-how that we will need to be able to establish a national kind of microbiome profiling platform, with the idea being to be able to get a snapshot or try to understand what is the baseline microbial diversity in the average South African community here, and how does this relate to various different factors. So we have an understanding, for example, from studies like the Human Microbiome Project, as well as the American Gut Project, of how these associations between lifestyle, diet, and environment may affect human health. But that understanding is lacking in the context of our unique environment in South Africa. And so the idea behind the CSIR Microbiome Mapping Initiative is to set up a foundation to generate that knowledge.
How does that tie in with the CSIR's mission? Because many people would ask, should the CSIR be involved in this mapping process, in the microbiome mapping process?
The core ethos of the CSIR and our mandate for the longest time has always been to improve the quality of life for all South Africans through technological and scientific innovation. And we strongly believe that this microbiome mapping initiative and this project very strongly kind of embodies that ethos, because it really exists at this nexus between a number of different fourth industrial revolution related technologies, such as big data, next generation sequencing, climate modeling, artificial intelligence. And it's at this nexus that we then combine all of these groundbreaking technologies in their own right to create this novel kind of resource that will have impact in terms of digital health and how we provide healthcare across Africa.
Doc, is it fair to say that a project of this level, I mean, you talked about Next Gen Health, but a project at this level would need wide-ranging expertise. Who do you bring on board to collaborate with on such a project to make sure that you kind of tick those boxes?
As biologists, of course, we have a very specific interest in the biology, but we also understand that in order to achieve what would be required for this platform, that we require a vast array of skills that we as biologists ourselves do not possess. And that, I think, is one of the advantages of being in an institute like the CSIR, where we have access to a broad array of cutting-edge technologies. So in this particular project, we've partnered with the air quality group at Smart Places, And they have come on board to actually assist us with establishing climate models so that we can begin to look at air quality and exposure for participants in the study at their home and work locations. And this, of course, will generate a large set of big data. And from the Microbiome Mapping Initiative itself, we're going to get samples from participants, and that's going to generate large sets of sequencing data. So we, again, need help in kind of interpreting the sequencing data. So we've brought on experts from the bioinformatics division at the University of Witwatersrand, who are collaborators on the project. And then there comes the great challenge of taking all of these huge data sets, so the climate, the next generation sequencing, and all of the anthropological data that we'll be collecting in terms of information from participants on whether they have a pet at home, because that influences your microbiome diversity, your diet, your lifestyle, how often you work out, all of this information, and then trying to say, how do we make sense of this? How do we know in all of these huge data sets what's relevant? And for that aspect, we've partnered with IBM South Africa, who have a vast experience in applying artificial intelligence and machine learning approaches to being able to gain deeper insight from large data sets. So through this multidisciplinary collaborative approach, the idea is that we will be able to kind of make sense and gain deeper understanding from all of these different data sets.
It sounds absolutely futuristic to me. It sounds like the plot or the basis of a great movie. How does one become part of this project? If you're interested in the mapping initiative and you want to volunteer, what is that process?
Okay, so that's great. We're actually going to be rolling out a recruitment drive on the CSIR campus within the next month or two, and hopefully we will be able to start recruitment in early January or Feb when people start coming back after the December breaks. And what's going to happen is that people can sign up online through a digital platform to express an interest in participating in the study. And what will then happen is that you'll be asked to submit a questionnaire online. And that's where we're going to collect all of the anthropological data about you in terms of, how often you work out, what do you eat, your medical history, in terms of like what diseases have you been exposed to, or have you taken antibiotics, all the things that could potentially explain why your microbiome is uniquely yours. That's the information that we will collect. People will then also be provided with a sample collection kit, and we're going to be collecting stool samples from individuals. These samples will then be processed. We'll extract the DNA. The DNA will be sequenced, and from the sequencing, we'll get an idea of all the different types of bacteria that are present in an individual's gut, and in what ratios. And then together with the guys from the climate modeling or Smart Places, we'll get the climate data, and the idea is then integrate all of this together to be able to start to understand in context of South Africa and South Africans what's driving these changes in the microbiome.
Will there be any benefit for those that volunteer with this mapping initiative? Will there be any benefits for them?
So because this is really a pilot study and the tech is something that we're developing, I have to be completely clear that this is not, we're going to take your microbiome profile and give you back a report that says to you, look, you've got to stop eating red meat today and you need to start eating more Brussels sprouts. No, this is purely, it's going to be a citizen science-driven thing at the beginning until we have enough knowledge to be able to make these assumptions. The idea is to gather this information. So what we will be doing, though, is providing people with a nice little report, which is similar to what's done in the American Gut Project in the US at the moment. So people actually volunteer for the study, they contribute their samples, and what they get back is a little report that says, this is the breakdown of the different bacterial species that are in your gut, and this is how, for example, your sample may relate to everybody else at the CSIR campus, or this can relate to, and we can break it down into different groups, like this is where you placed with everybody else that eats meat, or this is how far you are from a vegetarian.
So there are real benefits.
Yes. Yeah.
Second last question. We haven't touched on COVID-19 yet. How does microbiome profiling, the data extracted from, could help us potentially with the COVID issue?
That's an interesting question, and there's been a lot of interest around the microbiome and COVID-19 for a couple of reasons. One of them, which is really important, is that the receptor, which the COVID-19 virus uses to get into the cells, the ACE2 receptor, is present in lungs, and we know that that's why there are the issues with the infection. in the airways. But one of the regions in the human body that most highly expresses the ACE2 receptor is actually the human gut and the intestine. And we know that in a percentage of individuals who test positive for COVID, they present with a lot of gastrointestinal issues as well. So there's an hypothesis that there's definitely some strong link potentially between the exposure to COVID-19 and an impact on the microbiome. And what people are really trying to understand now is whether your microbiome profile at the beginning actually is causal or can determine whether you go on to develop severe COVID, or it actually has an impact in terms of determining how sick you get. But for us, and I think in terms of the microbiome profiling initiative and where this becomes more relevant, is the beauty of the microbiome, as I mentioned, is that it can act as an early warning system. So from data that's been accumulated already, we have an idea that certain microbiome profiles and shifts in the microbiome are associated with higher risk of disease A, B, C, and D. And one of the outstanding questions that remains now in terms of this current COVID climate is, what are the long-term implications of being exposed to COVID? And we know that recovery rates are high, but from data coming out from the UK now, we also see that even people who have recovered have gone on to display long-lasting symptoms. So people have long-term fatigue, people have developed things like depression, and other kind of disorders. And so this is where the microbiome potentially has the stage to really shine in terms of looking at, after COVID exposure, what was the impact on my microbiome, and how does this impact potentially translate to long-term effects of COVID exposure, and what diseases I will now be susceptible to that I probably wasn't before.
And talking about impact, how will a technology platform like this revolutionize both public and private sectors?
If we have to think a little bit ahead into the future in terms of, once we've established this microbiome profile or the mapping initiative and the platform exists, and we have enough knowledge to make the associations between healthy and diseased states in context of a South African climate, in terms of, let's just say, precision medicine, for example, and the provision of healthcare, for one, it becomes tremendously important, because a microbiome test becomes almost as important as a genetic test or as a blood test in terms of identifying what is the right therapy for you as an individual. So in that sense, it's really important in, one, ensuring that the right patients are matched with the right drugs, and ensuring that the state and medical aid schemes, etc., don't waste money on treatments that aren't going to work for people. In terms of the public sector, one of the things I mentioned was creating or developing this microbiome platform as a novel metric of health. And so when we think about accountability, this becomes really important. So when you look at things like poor service delivery in terms of water quality, or say a chemical plant has been put up next to a community and it's been there for the last 3 years, it doesn't help us to go in 20 years and say, geez, all of the kids in this community have gone on to develop asthma or cancer. What we want is to, within a year or 2 years, to be able to say, look, we've sampled everybody in this community, and based on their microbiome profiles, everybody has a higher risk of developing asthma in the next 5 years. We need to intervene now. And it's also, I guess, in that sense, being able to have a metric to hold people accountable for.
Wow, that sounds amazing. Dr. Jerolen Naidoo, thank you very much for your time.
Oh, thank you very much, Sam.